Hello, this is a video on working with transgenic organisms. What are transgenic organisms? They are organisms that have genetically altered genomes to carry transgenes. Most transgenic organisms are generated in the laboratory to address biological queries. However, constant exposure to transgenic organisms may pose potential hazards to human health. Let's talk about biosafety levels 1 and 2, or known as Level 1 Standard and Level 2 Standard. Level 1 Standard involves low-risk microbes that pose little to no threat of infection in healthy adults and without the use of special contaminant equipment, whereas for Level 2, pathogenic or infectious organisms that pose a moderate health hazard is involved. Next are examples of transgenic organisms commonly used in laboratories. The first example includes E. coli and phages. Working with such pathogens is vital to ensure effective applications of biotools in treatment, control, and prevention of ever-evolving threat of infectious diseases worldwide. Level 1 Standard is applied to non-self-transmissible vectors, whereas Level 2 Standard is applied to either self-transmissible or non-self-transmissible vectors. Secondly, the example is yeast, a non-pathogenic organism which poses no hazardous threats to other living organisms. However, they have developed deficiencies that necessitate a special feeding medium in a confined environment. Classification assigned is Level 1 Standard. Both primary cells and established cell lines are used in the lab. By determining the ability to create virus particles, biosafety level will be determined as follows. Level 1 Standard is applied to the production of cell line of HeLa with the combination of virus sequence in the cell line and have no result of viral particles. Meanwhile, Level 2 Standard is applied to both cell lines of primary mouse cells or cell lines of Epstein-Barr virus immortalized cells with the presence of a virus. The fourth example is viruses. Modified viruses are classified as the same or higher risk level than its biological counterpart. To produce replication defective virus particles, cell lines containing the missing viral sequences need to create virus particles are used. Level 2 laboratory standard are recommended. Last but not least is Agrobacterium. The disarmed strains of Agrobacterium belong to risk class 1 and is not applied to its wild type strains or Agrobacterium rhizogenes. Therefore, a basic level 1 setup are capable to contain a disarmed Agrobacterium in which the rules applied are similar with E. coli K12 strain. However, a basic level 2 containment are needed for a non-disarmed tumefaciens strain such as Agrobacterium rhizogenes. Much care should be taken in dealing with waste material from these transgenic organisms. The inactivation of biological waste and residues of such organism using appropriate medium is required, except for Agrobacterium. The waste should be decontaminated prior to discarding except for cell lines, which a different procedure will be assigned. The waste residuals should undergo disinfection process as well to avoid the leaking of cell material along with destroying proteins and enzymes necessary for the survival of the organisms. All solid and pasty materials such as the culture mediums and all liquids containing modified E. coli should be inactivated or decontaminated. The waste material also can be transported in a special container for infectious waste such as viruses to an incinerator suited for the incineration. Lastly, we certainly hope that this video will be useful enough to equip laboratory users to deal effectively with health and safety issues in relation to biological materials and toxins. Thank you for listening.